and welcome to another bio lesson to go. I'm Mr. Dove, and today we'll be looking at the whole process of photosynthesis. Photosynthesis is the process by which plants and other producers are able to take the energy from the sun and capture it into the molecules of a carbohydrate, like glucose. The whole process of photosynthesis can be summarized using an equation. We have carbon dioxide and water going in, and we're going to use the energy of the sun plus some plant enzymes to store that sun's energy in the bonds of glucose and produce some oxygen as a waste product. When we look at a summary equation like this, the items that are on the left side of the equation are the reactants. Those are the things that are going in, that are going to be reacting together to produce our products. You can think of it sort of like baking some cupcakes. The reactants are the different components that you're putting together, the parts of the recipe. Now, we're going to need some energy to be able to catalyze that reaction. So in this case, instead of sunlight, we're going to be cooking with a stove. And our products are going to be our cupcakes. So what goes in has to come out. That's why our equation is balanced. It has these coefficients which allow us to show that what is going in is also coming out, because matter can't be created or destroyed. The process of photosynthesis occurs in the chloroplast of plant cells. Chloroplasts are actually a pretty complex organelle, thought to have been derived from an early prokaryotic bacteria that could do photosynthesis. We find that our chloroplast is a double-membraned organelle. It has an external membrane, and it has a series of internal membranes called thylakoids. When we have a whole stack of these thylakoid membranes, we call them grana. In addition, surrounding these membranes, there is a fluid material called stroma. We can see that photosynthesis occurs in two kind of steps or stages that are happening simultaneously, but to better learn about them, we're going to take them kind of in a step-by-step fashion. The first step or stage is going to be called the light reaction, and then the second we'll be looking at is called the Calvin cycle. The light reaction, the job of it is to take the energy from the sun and convert that into chemical energy. So that's a really important process. So we know it's the light reaction because we're going to be using sunlight, and then we're going to be converting that into chemical energy. The light reactions are going to occur in the thylakoid membranes, the stacks of membranes called the grana. Within the thylakoid membrane, there's a green pigment called chlorophyll, and there's other molecules that are arranged in clusters that are called photosystems. And these photosystems, their job is to absorb solar energy. So you can kind of think of these photosystems sort of like a miniature solar panel. So what happens is light is going to strike that photosystem. And there are certain parts of this photosystem molecule which has electrons that can be excited by that light energy. And those electrons, they are moved to a higher energy state. They move into a different shell as they absorb that energy. And so this action is actually converting the sun's energy into chemical energy. Now, it's not just any wavelength of light that's going to excite those photosystems. It's kind of like Goldilocks and the Three Bears. We need just the right wavelength. Now, chlorophyll is going to use the part of the light spectrum which we call the visible light spectrum. All the different colors of the rainbow. Red, orange, yellow, green, blue, and violet. Now, the chloroplasts are going to really enjoy absorbing light in the red and blue spectrum. That's what really excites those reaction centers. But green light is actually reflected back to our eyes. That's why we're able to see plants as appearing green. So the plant is not going to use green light very much for photosynthesis, but instead it's using the other colors of the spectrum, especially the red and the blue. Now there are two photosystems that are involved in the light reaction, Photosystem I and Photosystem II. You might notice that Photosystem II comes before Photosystem I, and that has to do with how it was discovered. Initially, Photosystem I was discovered, and then they found out there was another photosystem, but it wasn't until later that they found out that the Photosystem II occurs before Photosystem I, and they just kept the names the way they were. So these photosystems are connected by a series of special molecules that can transport those electrons, and as those electrons move through that series of electron transport molecules, it's actually able to extract that energy and convert that into more useful forms. So photosystem 2 is going to excite some electrons when light hits it. Those electrons are going to be used to make ATP using an enzyme called ATP synthase. We know it's an enzyme because it ends in ase, and its job is to synthesize ATP, very much like ATP was synthesized in cell respiration. Additionally, so once we get to the end of that first electron transport system, the electrons are going to have been moved to a lower energy state. So once those electrons reach photosystem 1, they get re-excited, and then they're passed to an electron carrier that's going to then be able to take those electrons to another part of the photosynthetic process. Here, our electron taxi cab is going to be NADP instead of NAD like we saw in cell respiration. NADP stands for nicotinamide adenine dinucleotide phosphate. So here's kind of a little cartoon that shows what's happening. So our sunlight hits photosystem 2, which excites electrons. As they move through electron transport, that's going to help facilitate the production of ATP using that enzyme ATP synthetase. Because we're making ATP in the presence of light, this is oftentimes referred to as photophosphorylation. Now, once we reach the end of that initial electron transport, that electron is going to be lower in energy. So they're going to be re-excited with some more light, and then it's going to move down a shorter electron transport and be transferred into ATP to be carried down the way. Now, unfortunately, the photosystems don't have an endless supply of electrons. So what happens is that water is used to replace those electrons. Water molecule is going to be split using light, a process known as photolysis. When you split water, we're going to get some high energy, or we're going to get some electrons that we can put back into the photosystem. We're going to have oxygen and then some protons, some H's, that we can send along with our electron carrier, that NADP, when it becomes NADPH. So if we wanted to summarize the process to make it easy to write down for our notes, first, light was exciting the electrons of the photosystems. First, we see photosystem 2 taking those electrons down an electron transport system to be able to produce ATP with the help of an enzyme, ATP synthetase. Then we have to re-excite those electrons using photosystem 1 and a bolt of light energy, and it'll move down a short electron transport chain to be carried by NADP as NADPH into the next step of photosynthesis. And then finally, we're going to need to split water to be able to replace those electrons. And as a byproduct, we're producing oxygen, which is what we're breathing. So in reality, what we're breathing as oxygen used to be a part of a water molecule. So if I wanted to illustrate this in a real kind of simplified cartoon, here's how we would start. So the sunlight is going to hit those photosystems present in our grana, because it's got the green chlorophyll, so the grana is green. That light energy is going to be transferred into chemical energy in the form of ATP and NADPH. To replace those electrons that were transferred on, we're going to split some water by photolysis, and that's going to result in our production of oxygen. So at this point, we've taken our light energy and stored it temporarily in as chemical energy as NADPH and ATP. So you might be wondering, well, why don't we just stop here? I mean, we've got chemical energy, and ATP is useful for cells. Well, there's a problem. ATP and NADPH is relatively unstable, so they don't store energy for a long period. You can't dissolve them and transport in tissues. And they're not very useful as raw materials to make other things, so we have to move on to the next step or stage, which is the Calvin cycle. The Calvin cycle is named for one of its discoverers, Melvin Calvin. The job of the Calvin cycle is basically as a sugar factory. It's going to make sugar using the carbon dioxide atoms, the hydrogen that's coming from the light reaction, and the energy that was stored during the light reactions. Now, the Calvin cycle is happening in the liquid part of the chloroplast, the stroma. Now, this might be difficult because stroma is very, very similar to that opening in the bottom of the leaf called the stoma. Please do not get these mistaken. The way I kind of remember the difference between the two is since the Calvin cycle is happening in the stroma, I kind of imagine a little guy by the name of Calvin swimming in the stroma. So he's doing the backstroke in the stroma. Stroma. He's doing the backstroke, and that might help you keep them straight. Now, sometimes if you're doing research or checking in your textbook, that the Calvin cycle can be referred to as the dark reactions. Now, the reason why they're called the dark reactions is not because it happens in the dark, but because the process is occurring in the stroma, there's no chlorophyll, so it can't use the light directly. So a better term is perhaps the light-independent reactions. So in order to gain energy to process and create that sugar, the light reaction is serving sort of like as the electricity generator for our sugar factory. So since it can't use the sunlight directly, it needs to get some help by utilizing the light reactions. So in the Calvin cycle, we're going to take carbon dioxide and we're going to use the energy that was from ATP plus the high energy electrons and the protons that are coming from the light reaction to produce our glucose. So our first step is we need to get the carbon dioxide into the Calvin cycle. Well, this is done by an enzyme called Rubisco, which stands for ribulose biphosphate carboxylase. We know it's an enzyme because it ends in ase, and it's going to carboxylate. It's going to put the carbon with ribulose biphosphate. Ribulose biphosphate, or RuBP, is a compound that's present in the stroma. So the enzyme helps to kind of put them together. You want to think of it sort of like a marriage. Carbon dioxide is going to be married to RuBP, and the officiator of the marriage, the pastor, the rabbi, is going to be the enzyme, ribulose biphosphate. So carbon dioxide is fixed. It's brought into the Calvin cycle with by connecting it to RuBP with the help of the enzyme Rubisco. What that does is that creates a real simple sugar called 3-phosphoglyceric acid. Unfortunately, this is not going to be very useful for the plant, so we need to further transform it. So in order to do that, we're going to use that energy from ATP, the electrons and the protons, to be able to convert that PGA into a very useful sugar called phosphoglyceraldehyde, or PGAL. PGAL is then going to be used to make glucose and other useful molecules for the plant. It will take 6 carbon dioxide molecules to be brought in to produce one glucose molecule. So our 3-PGA is going to be converted into that useful sugar PGAL, which can be used to make glucose. Now, some of this glucose is actually going to be used by the plant itself for food, and some will be stored, and that could become food for consumers. Now, if we took all the PGAL and made glucose, this would no longer be a cycle. It would just go and then stop. So we need to be able to keep the cycle going. To keep the cycle going, we hold some of the PGAL back, and we use that to produce glucose, so that we can, to produce more RuBP, so we can keep the cycle going. We have to keep making RuBP because it's a limiting reactant to this Calvin cycle process. We don't have to remake Rubisco all the time because it's an enzyme, and enzymes are reusable. So our PGAL, some of the leftover PGAL is going to be recycled with an input of a little bit more ATP, and that will make more RuBP so that we can fix more carbon dioxide and keep the cycle going. So if we wanted to summarize this process for our notes, first thing we need to do is fix the carbon dioxide with RuBP using the enzyme Rubisco and make that real simple, kind of not very useful sugar, 3-PGA. Using the energy stored from the light reaction, we're able to convert that PGA into a more useful sugar, PGAL, which then can be used to make glucose and recycle RuBP to keep the cycle going. So here, for our final summary, this is a really good diagram. It's at the bottom of your notes. You'll be able to kind of follow along, and this will be really helpful on your test. So we begin by the light reaction, capturing the light energy, converting that light energy into the stored chemical energy of ATP, and high energy electrons being carried by NADPH. To replace those electrons lost, we're going to split water, producing oxygen. In the Calvin cycle, we're going to take carbon dioxide and fix it with the help of the enzyme Rubisco. Using the energy that was stored from the light reaction, we're going to be able to produce that all-important carbohydrate sugar, which can be used by the plant or to be food for you and me. I hope these notes were helpful and simplified enough that you've really beginning to understand the process of photosynthesis. Please go back and watch it again to really make sure that you have a full understanding of this very important process.